Okay, so we left off at the end of 15.4, so we're going to do 15.5 here. So let's look at the title. It says some inheritance patterns are exceptions to the standard chromosome theory. So there's more exceptions. There are two normal exceptions to Mendelian genetics. One exception involves genes located in the nucleus, and so that's what we've been talking about so far. Everything sex-linked, all that autosomal, all of those genes are found in the DNA in the nucleus. But if you recall from in the cell chapter, that's not the only place that DNA is found. So the other exception involves genes outside the nucleus. Now, can you remember what organelles have DNA in it besides the nucleus? You should have thought that it could be in chloroplast or in plant cells or in our cells or in all cells that are eukaryotic is the mitochondria. Both of those have DNA. So let's look at this. So let's look at the exception in the nucleus. So in the nucleus, this is genomic imprinting here. So let's look at what genomic imprinting is. For a few mammalian traits, the phenotype depends on which parent passed along the alleles for this trait. So just for a few traits. So most of the time when we've talked about if somebody is big R, little r, for instance, it doesn't matter if you got the big R from mom or the big R from dad or the big R from dad and the big R from mom. It doesn't really matter. You still end up showing the dominant trait, and it doesn't matter who you got it from. And so that's for most cases, but there are a few cases where it depends on, you get a different phenotype based upon whether mom or dad gave you the big R or the little r. So such variation in phenotype is called genomic imprinting. So genomic imprinting involves the silencing of certain genes that are kind of stamped with an imprint during gamete production. And so this is usually with a methyl group. Let's add methyl groups to the cytosine nucleotides of the DNA. Adding methyl groups to the nucleotides of the DNA of the gene, that leads to the silencing. What does that mean? The gene doesn't work. You can't make the protein. Even though you have the gene, the addition of methyl groups kind of like turns the gene off, so you can't make the protein, and therefore you don't get the trait. So let's flip over and go through an example of this that they figured out in mice. All right, so here's your normal. It says a wild type mouse. So that wild type, remember, is normal, most common. And so here's the wild type mouse's normal size. And here's the pair of chromosomes that determine the size of the mouse. So the gene we're looking at is this gene right here. So this is the chromosome made out of a piece of DNA. So there's a piece of DNA, two pieces of DNA. Paternal, remember, came from dad. Maternal came from mom. So each chromosome is made out of two pieces of DNA. Notice that this mouse, this example here, has two normal genes. All right, so there's the normal, and IGF is insulin-like growth factor. That's what they stands for. So the growth factor, just think, this is something that this gene codes for. You make this growth factor that causes normal growth in the mouse. So that's why the mouse is normal size. All right, and here's the second. So these guys are, this mouse is homozygous, has two normal genes, and therefore grows properly. Now notice it says expressed and not expressed. What they figured out is that only the gene, even though mom gave this mouse a normal growth factor gene, this actually is turned off. That's what it means by not expressed. This one is the only one that's on. What they found is that no matter what, even you have two genes that determine how big you are as a mouse, but only the gene that's on the chromosome that you got from your dad, your paternal chromosome, is actually one that's expressed. And so let's look at how they kind of studied that. So they studied normal and dwarf size mice, and so normal size versus dwarf. And what they noticed, and they studied the DNA, so here's the chromosome pair. Each one is made out of a piece of DNA. Notice here, I'm going to just use the same color here. They got a normal gene here. So here's your normal gene. And then they got a mutant gene. I'll use pink here. So they got the mutant gene. So something happened to change the order of bases, A's, T's. I'm doing pink here. It's hard to see, I know. I'm doing pink there. And that's your mutant. And so this is a mutation. So they're heterozygous, where they have a normal gene and a mutant gene. And look, they appear normal size. And so, but then if you look at here, the dwarf mouse, and look at their chromosome pairs from mom and dad, they also inherited a mutant gene, but instead the mutant gene came from dad, and they also inherited a normal gene, and the normal gene came from mom. And so, in looking at this, they are genetically identical, this mouse and this mouse, where they each have a normal gene and a mutant gene, normal gene and a mutant gene. And unlike other things where it doesn't really matter, a lot of the traits that we've talked about doesn't really matter if you got the big R from mom and the little r from dad or vice versa. This seems to matter. What we seem to find is that this mouse was normal, and that normal gene was on the paternal chromosome. This gene was dwarf, genetically identical, but on the paternal chromosome was the mutant gene, so therefore they're not making the insulin-like growth factor, so therefore they don't grow properly, and so you get a different phenotype. This is one of the things that they found to support genomic imprinting, is that this is one of the genes that does that the maternal, whatever is on the maternal chromosome here, these guys were methylated, and the genes is turned off, and so it's as if you don't have it. And so therefore whatever's on dad's chromosome that he gave to you, or in this case the mouse, that's what you make in terms of the protein, and therefore that's the trait that you have. And so same genotype, different phenotype based upon which parent gave you or gave the mouse the gene. And that's genomic imprinting. And so one thing I want to point out, though, is that now this mouse and all of his cells, or her cells, they're expressing the normal gene, and so therefore they're making the protein and growing normally. Now when this mouse goes to make their gametes, their egg or their sperm cell, depending upon the gender, you reset. And so therefore you reset the paternal and maternal chromosomes, and then the sperm fertilizes the egg, and then once you get a zygote, then the new paternal chromosome is the one that is active, and the maternal one is methylated again, so for the babies. All right. And then lastly, so this is in the nucleus. So back to the beginning here when we said one exception involves genes in the nucleus. So let's put this in nucleus. That's genomic imprinting. It happens with the DNA in the nucleus. And then the other exception involves genes outside the nucleus. So this is inheritance of organelle genes. So this is outside the nucleus. And we call genes that are located outside the nucleus extranuclear genes. nuclear genes. They're genes found in organelles in the cytoplasm. So example would be mitochondria and chloroplasts. The inheritance of traits controlled by extranuclear genes depends on the maternal parent because the zygote's cytoplasm comes from the egg, not the sperm. So therefore you get in plants, most of the plastids or chloroplasts come from the parent that donated the egg, so the quote-unquote mom plant. And same thing with mitochondria, even for us. So the zygote cytoplasm comes from the egg. The first evidence of extranuclear genes came from case studies on the inheritance of white or yellow patches on leaves of an otherwise green plant. And so in this case here, the genes were inherited in the chloroplasts. So therefore, when the sperm fertilizes the egg is where all the chloroplasts come from. When the sperm fertilizes the egg, it doesn't give the chloroplasts that were part of that sperm. All right, and so it's from the mom. So any genes within the chloroplast came from the mom. By the same token, in us, the same kinds of things happen with the mitochondria. Some diseases affecting the muscular and nervous systems are caused by defects in mitochondrial genes that prevent cells from making enough ATP. So remember mitochondria, that's cellular respiration. Remember, cellular respiration occurs there, which makes ATP. This is the egg cell. All right, it has the nucleus and it has all the mitochondria in it and so on. And here's your sperm cell and here's the flagella. Most of the mitochondria are down over here by the quote-unquote tail or the flagella. And so when the sperm, let's label this sperm, comes to fertilize the egg, it just donates its DNA. It doesn't donate its mitochondria. So once fertilization happens, then you get your zygote. And so in this case here, it'd be 46 chromosomes, but all of that mitochondria came from mom. So what we're saying is any genes in the mitochondria come from the mom, and then you divide by mitosis, and so all your mitochondria, basically in all your cells, you inherited that from your mother, all right? And so let's add a few things. I'm going to add a few bullets here. Let's add here, most mitochondrial genes help make the protein complexes of the electron transport chain and ATP synthase in cellular respiration. So hence, if you have mutations in that gene, you may have issues with doing these things and making, therefore, the proper amount of ATP via cellular respiration. So an example of a disease is mitochondrial myopathy. So let me give you some examples of characteristics. So if they're not making enough ATP because of a mutation, they have muscle weakness, maybe some intolerance of exercise because of the lack of ATP for energy, and they can either also have some muscle deterioration. All right, and then lastly, last bullet here, There are some mitochondrial mutations that have been linked to some other things. Also thought to be involved in some cases of diabetes. Heart disease, and Alzheimer's. All right, and so obviously lots of research being done on that. So that's another unique thing, all right, is so we have lots of different types of rare genetic disorders that are due to not mutations in the DNA in our nucleus, but they're due to DNA mutations in our mitochondrial DNA, and all of those collection of disorders usually affect, as we said, the muscular or nervous system. All right, and so this is where I'm going to stop for the video, so it's nice and short and sweet. The next class period, we'll go back and we're going to look at 15.2 because we skipped that. All right, so we'll go back and do that.